Okay, I think, Iman, if you are happy, I think we should probably get started. First of all, I just want to welcome everyone to this Desmond Tutu HIV Foundation event. It is entitled Vaccines and How Getting to Know Them. And that is, of course, around SARS-CoV-2 and COVID-19 as the disease. And I'm sure all of you have been hearing various things about vaccines in the media. The Desmond Tutu HIV Foundation, as a trusted partner, wants to come alongside you, recognizing that we can all follow good advice and do the best possible things for our own health if we have good information and we know what is the best options available to us. And since COVID-19 has only been around with us since January of 2020, we have had literally, 13 months to come to terms with this new disease. Luckily, there are vaccines available around the world. And between now and when South Africa will be rolling out vaccines more generally to the general population, we will undertake a series of these webinars to try to familiarize you with the science as it unfolds. And it really is a day-to-day exercise. I'm sure all of you have been hearing over the weekend about variants and vaccine efficacy and a whole lot of other subjects. We will unpack those over the next few webinars. So we won't cover everything today, of course, but we will make a good start. And I look forward to hearing what your questions are, hearing what your suggestions are. And please do let us know if there's a particular subject you'd like us to cover in the next few weeks to months. So today we have a great lineup. We're going to start with Dr. Katherine, who will be telling us basically about vaccines in the most fundamental way. We're starting at the basics. We'll then go on to Dr. Sheetal, who will be telling us which COVID vaccines are approved in the world today and how they specifically work. And then we're going to turn to Ntando and Nombeko, who will cover some community perspectives, what people are hearing on the ground, what are some of the ideas that are being passed around, and we'll try to give some evidence-based to what you are hearing in the communities, what we are hearing in the communities in terms of what we know is unknown, what is true, and what is truly not true. That is what we're hoping to try and sort out for you today. So without further ado, I'm going to hand over right away to Dr. Katherine, and I'm going to remind you, please, to record your questions in the Q&A box. And we will get to that towards the end of the webinar. We're not going to keep you longer than an hour. So send your questions as soon as possible, and we can deal with some of those even as we go. So with that, I'll hand over to Dr. Katherine. Thank you.
Okay, thank you, Linda-gail. So I'm Katherine, I'm a doctor, and I worked on the J&J study, and I'm going to tell you a little bit about vaccines today. So just before we can even start talking about vaccines, we have to talk a little bit about our immune system. And what I thought was very helpful is to imagine a tree as our immune system. So we start off with the bark, and that is like our mucous membranes, and basically anything that stops bugs from getting in. And then we go into our adaptive and our innate immune systems, and I'm going to talk about those a little more. Okay, so the innate response is our first line of defense against bugs. It's non-specific, it can't prevent infection, so it does allow invaders to come in. It's comprised of these cells called natural killer cells. Activation of the innate response is required before our adaptive or our learned response can happen. It doesn't have memory. And the way these natural killer cells work, they're quite cool. They basically jump on top of bugs and pop them. Then the adaptive immune system. So another word for adaptive is acquired. And this is specific. So it's specific for different kinds of antigens. So we will have a specific response to a cold virus, to a measles virus. It takes several days to become protective. It develops throughout your life. So we know that when we send our children to creches, when they're babies, they are sick all the time. And as we age, so our immune system is primed to be able to deal with anything that we see. And there are two components. There are a cellular component and a humoral or an antibody component. So the humoral or antibodies are made by cells called B cells. And it takes about 2 days after infection for your body to start making antibodies. But usually it needs about 2 weeks for full effect. And that's important when we're looking at COVID vaccines. So we know that from about 2 days after you get the vaccine, you'll start producing antibodies, but the full effect is only felt a little bit later. Antibodies have 3 functions. They can neutralize or stop viruses. They can eliminate the virus through something called opsonization, which I'm going to show you how that works. And they can also sensitize the immune system to engage other functions. So antibodies can also prevent infection, and they have memory. So this is just a little graphic to show how antibodies work. So you can see a virus there in the middle, and the antibodies bind and then destroy the virus, and that's the first way they can prevent infection, and that's called neutralization. And the second way that they can prevent infection is that they can recognize an invader. They can bind to it, that you can see there, and it's almost like a red flag to a little cell that's going to come along. The cell's going to pull the virus inside itself, and then it's going to kill it. It's going to destroy it. Okay, so now we're going to look at the cellular component of the immune system. Cellular response involves 2 types of cells. One are called helper T cells, and then they are the cytotoxic T lymphocytes. They have memory, and they can be trained by vaccines. So we're not only looking to create antibodies, we're also looking to boost these kinds of cells to help us fight illness. So CD4 cells, a good way to remember them, they are like megaphones. They call the rest of the immune system to come and help when they see something. And the thing that they call are the CD8 cells, which are the killers. They're like the great white sharks. They are very, very effective at eliminating bugs. So how does the adaptive response work? Basically, the T cells are looking the whole time. They are surveilling your body. They check other cells. They check whether they can see anything that's infected or abnormal, and then they destroy these abnormal cells. So just in summary, the cellular response, it can't prevent infection. T cells are activated when cells become infected. They can eradicate an established infection. They have memory, and they can be primed by vaccination. Whoops. So then if we look at actual vaccines, preventative vaccines have been used for decades around the world. We know the most commonly used in children. They are very safe when manufactured and used properly. They are very cost effective compared to treatment. If we think about a disease like polio, imagine how much money goes into treating somebody who's paralyzed compared to preventing that infection. We know that we've eliminated smallpox, and we have also eliminated, in 2019, the WHO announced that we've actually eliminated 2 out of the 3 types of polio. So then this table is just to look at how long it usually takes to develop a vaccine. If you look at the top, Haemophilus influenza has taken almost 100 years to develop that vaccine. We know that we've been working on the HIV vaccine for the last 30 years. And one of the questions I think people in community are asking is, how did we develop these new vaccines so quickly when previously it's taken such a long time? And I'm going to briefly go into that. Then we can also look at this is particularly for the United States, but it also applies to Africa. Basically, in the first column, we look at how many cases we had of illnesses before a vaccine was introduced. And something like polio, we can see 16,000 cases of paralytic polio pre-vaccination. And if we look at 2008, there were 0 cases. So it's 100% prevention. And I think we need to reflect on this, because I think vaccines have been so successful that we have forgotten how much devastation they used to cause. And we really need to be mindful of how far we've come. So then, what are vaccines? They basically are substances that teach your body how to recognize and fight invaders. The body recognizes a virus, it sounds an alarm, the fighter cells and the proteins go into action, and the goal is for the virus to either be controlled or neutralized. So then we've been developing vaccines for a long time. There have been traditional approaches for developing vaccines. One of them is called a live attenuated vaccine. And basically what happens here is that we inject an actual virus into somebody. So we would inject a measles virus, but it's been attenuated or weakened so that it can't actually cause the disease. Another strategy is to inject a whole virus that's been inactivated or killed. Examples of this are the whooping cough vaccine and the rabies vaccines. And we are still using both of these traditional approaches for developing COVID vaccines. What's exciting is that we also have new ways of developing vaccines. And to understand those, we need to go back to what SARS-CoV-2 looks like. So if you look at the picture, you can see a human cell in orange, and then you can see the yellow coronavirus, and it's surrounded by these spike proteins. And then on the right, you can actually see what the spike protein looks like, and that is the target for most of the vaccines that are being developed. So then just quickly to talk about these new mRNA vaccines, basically how they work is that we inject messenger RNA into a person. Your body then uses the mRNA almost as a recipe to create the spike protein found on the coronavirus. Your body then learns, it looks at the spike, it triggers a whole immune response, and basically you teach yourself how to deal with the virus. So these are very new. They're made by 2 companies called Pfizer and Moderna. What's exciting about these vaccines is that as soon as you know the sequence, the genome of the virus, you can start immediately to develop these mRNA vaccines. So this is one of the reasons why we could develop the vaccine so quickly. The big problem with these vaccines is that they're very fragile and they need to be kept very, very cold. Another very new and exciting way of developing vaccines is called the viral vector vaccine, using an adenovirus. And basically what we do here is we have the spike protein, which is compared to a package, but the package can't deliver itself, so it needs a FedEx truck, so it needs a vector to deliver the parcel into the cells, and the vector that we use is called an adenovirus. This adenovirus it's a type of cold virus, and it's been made weaker so that it can't actually cause a cold in the person, but it purely acts as a truck to deliver the spike protein into the cell. So a problem with these viruses is that adenoviruses are very common, and you don't want to put this vaccine into somebody who's seen an adenovirus before, and then the vaccine is neutralized before it's even had a chance to work. So what scientists have done is they've found adenoviruses that don't usually cause disease in humans, something like an adenovirus 26 that J&J uses, or another strategy is to use a virus that infects a primate, like the chimp adenovirus that's used in AstraZeneca. And also the Russian Sputnik vaccine is also using an adenovirus vector. What's exciting about these vaccines is that you get an immune response to both the vector and to the spike protein insert. So it's almost like a double response. Importantly to note, because I've heard concerns about this, that they don't interact with our DNA. And also very exciting with these vaccines is that they're a lot more stable than the mRNA, and they don't have such demanding cold chain requirements. In summary, vaccines have been around for hundreds of years. They've prevented much disease, death and suffering. They have this effect where they don't only protect the person who has the vaccine, but they can protect other vulnerable members of society through this concept of herd immunity. They undergo rigorous safety testing before being introduced and are constantly monitored for side effects after introduction. And they have this exciting potential to completely eliminate disease. Thanks.
Thanks, Katherine. I think that was a brilliant overview. I'm sure it's also thrown up lots of questions. Don't worry. We're quite happy if you come back week after week. We'll continue. This is tough science, as the minister said last night. Sorry about the hard science, but it is important for us to grapple with how the immune system works, and it is a wonderful, wonderful part of our bodies. Because the vaccine and our immune systems are working together, right? They're in sync to beat these diseases. And so it's important you understand the one in order to get the other one. So don't panic if you're feeling a little bit lost. This will take time. I've been at this for 25 years, and I am not sure I have completely got a handle on all of it. So come along on the journey with us, and we promise it will get better over time. Now I'm going to hand you over to Dr. Sheetal because she's going to add a little more on the layers going forward. Over to you, Sheetal.
Hi, everybody. Thanks, Katherine, for that presentation. And that really leads nicely into my presentation, which is going to be on which COVID-19 vaccines are already approved in some countries around the world, and a quick overview on how they work. So I just wanted to start by saying that we know vaccines. We know vaccines that can work. We have a very well-established childhood immunization program here in South Africa, and this really gives us a good platform for the COVID-19 vaccine that we hope to be able to use soon. So what is the COVID-19 vaccine? In the previous presentation, you did see the coronavirus and how it looks, the structure of the coronavirus. And I wanted to particularly point out that spike protein is what we are targeting. And those little spikes that are around the coronavirus are what our vaccines are wanting to work against in order for us to develop an immune response. So the COVID-19 vaccine is intended to provide immunity against the disease called COVID-19 that is caused by the virus SARS-CoV-2. In general, vaccines contain weakened or inactive parts of a particular organism that triggers an immune response within our bodies. And this weakened version, importantly, will not cause the disease in the person receiving the vaccine, but it will prompt our immune system to respond. And we know that some vaccines require multiple doses, and this is a way of training our body to fight the disease and to build up some memory against the disease, the organism, so that if we're faced with it in the future, we're able to fight it. And really understanding how these COVID-19 vaccine work will help us trust the vaccine and also allay any fears around them. So you've already seen an antibody slide, but this is what we want. We want antibodies that can bind to the spike protein, and then we can kill this virus. And that's what our vaccines are trying to do. So let's quickly look at some COVID-19 vaccines around the world. There are 6 vaccines that are known and approved, and the first 2 are the Moderna and the Pfizer BioNTech vaccine, which are the mRNA vaccines. Then I'll talk about the viral vector vaccines, which is the Oxford AstraZeneca, Johnson & Johnson, and the Gamaleya vaccine, which is the Russian vaccine. And then there is a whole inactivated vaccine, which is the Sinovac, which is a Chinese developed vaccine. And then the last vaccine that I'd like to talk about is the Novavax, because we've heard a lot about it over the last few days. I just touch on that. That vaccine is not approved. It's still undergoing approval. On this slide, we know now that the Johnson & Johnson vaccine efficacy is 66% globally, and it protects us against severe disease 85% of the time. So as of last night, the total vaccination doses given globally so far for COVID-19 is 128,347,297 in 83 locations around the world. So it's quite significant. Okay, so the first group of vaccines are the nucleic acid vaccines, and Dr. Katherine did touch a bit on how these work. So mRNA vaccines are the Moderna and the Pfizer vaccines. These vaccines are being used in the UK, the US, Canada, Switzerland, and South America. And mRNA is a new technology. What mRNA does is it teaches our cells how to make that harmless piece of protein that triggers an immune response. And in this case, it's the spike protein. Importantly, the mRNA cannot cause infection. The mRNA is just a bit of genetic material that is given to us via injection, and it is encased in a fat cell or lipid cell. And this is given to us, and our cells read the mRNA like a recipe again. They don't interact with our body's DNA in any way. And our body learns to develop an immune response against that spike protein. So these two mRNA vaccines, the Pfizer and the Moderna vaccine, they're a bit complicated for our setting because it has to be stored at quite low temperatures. And another thing is that they have to be given 2 doses 28 days apart. Okay, the next group of vaccines is the whole inactivated vaccine. So this vaccine uses a whole inactivated virus. What happens is that the coronavirus is killed, the genes are killed in a lab using radiation or heat or chemicals, and then these inactivated viruses are grown, and this is injected via a vaccine. Our body then takes up this inactivated virus or dead virus and then develops an immune response and learns how to fight this virus. It's been approved in China and the United Arab Emirates, and it's used there. It can sound a bit scary, but I wanted to say that we have used whole inactivated vaccines before, and that is in the polio vaccine, hepatitis A, and the rabies vaccine. Importantly, again, these vaccines can't cause infection. And then there is this group of viral vector vaccines. So what viral vector vaccines do is they use a modified version of a different virus, in this case, usually adenovirus for the coronavirus vaccine, to deliver important instructions to our cells. And that important instruction is how to develop antibodies to the spike protein of the coronavirus. The viral vector vaccines do not cause illness of the viral vector that is being used, so it won't cause, for example, if adenovirus is being used, it won't cause adenovirus disease, and it would not cause COVID-19 disease. And there again are examples of viral vector vaccines that are being used currently, and those examples are the Ebola vaccine, RSV, malaria, and the Zika virus. So the first viral vector vaccine I'll talk about is, very briefly though, is the Sputnik vaccine, which is the Russian vaccine. It's currently being used in Russia and Europe and Argentina. And again, it's using a viral vector. So the vector that it uses is the adenovirus, and AD26 and AD5. It's mostly used in Russia. It also requires quite significant freezer storage, and it's given 2 doses 3 weeks apart. And then the AstraZeneca vaccine, the Covishield vaccine. We've all heard about this extensively in the media, and we know that this is a vaccine that has been brought into our country. And how this vaccine works is also a viral vector. It uses the chimpanzee adenovirus, and adenovirus is a virus that causes a common cold or flu-like symptoms. What happens is the researchers weaken the adenovirus in the lab so that the virus cannot replicate in the body. And then there's a bit of DNA inside, or a little recipe inside the adenovirus that gets injected via the vaccine that teaches our body how to make a spike protein. And then we develop that immune response that Dr. Katherine talked about. This vaccine has been approved by the South African Health Products Regulatory Agency for emergency use in South Africa. But as we've heard, it hasn't, as we've heard last night, it's not very effective against our South African strain. And that brings me to the Johnson & Johnson vaccine. So this is very similar to the AstraZeneca vaccine in that it is a viral vector vaccine. It uses a different viral vector called the adenovirus number 26. And again, very similar. The researchers weaken the adenovirus in the lab so that the virus can't replicate in the body. And then this teaches our body how to make the spike protein so that we develop that strong immune response. This vaccine is still undergoing the approval process. The results were only released a couple of weeks ago. And then the last vaccine I wanted to talk about is the Novavax vaccine. Like I said, it's not yet approved anywhere, but it has been spoken about. It is a protein subunit vaccine. Now, this type of vaccine uses a synthetic protein. So it's a lab-made version of the SARS-CoV-2 spike protein that is mixed together with an adjuvant, which is an adjuvant is basically a substance that will stimulate our body's immune system response. And this synthetic protein is given via a vaccine. It's not yet licensed for COVID-19, but again, there are other protein subunit vaccines that we know and that have worked. And that is the diphtheria vaccine, tetanus, and the meningococcal meningitis vaccine. So in conclusion, we've seen that there are many COVID-19 vaccines that are in various stages of approval. There are different types of vaccines and they all work slightly differently, but again, they're all working toward the same goal, and that is to build immunity against COVID-19 disease. Understanding how these vaccines hopefully will enable us to trust them and allay any fears about these vaccines. We know that vaccines in general have been proven to be effective in preventing disease, and the vaccines that I've spoken about, apart from the mRNA vaccine, the whole inactivated vaccine, the protein subunit, and the viral vector vaccine has all been used to target other diseases before. And it is very important that these vaccines cannot cause COVID-19. They don't change the body's DNA in any way, and they don't contain any microchips. Thanks.
Great. Thanks, Dr. Sheetal. That was brilliant. So I think you guys have had a wonderful foundation. I'm sure there are many questions in your minds. Don't forget to just post them in the Q&A. And we are now going to hand over to Nombeko and to Ntando and hear from the community. So over to them both. I'm not sure if they're doing a duet or in sync.
Hi everyone, I'm going to start and then Tando is going to follow. I am very excited to be part of this panel. I'm checking the number of people who are in this webinar. So I'm adding, I've got mothers and children who came for the REVAX session and we discussed what is going to happen and they have decided to join voluntarily. So altogether, we have 60 plus pre-committee staff members. So we've got 90 people here that are also part of this meeting. So I'm going to continue. Oh, okay. It's 16, and then there's also one participant that is with ensemble study. that also has decided to sit and be part of the session because she had questions that were answered last week. Okay, so we're talking about the committee concerns about COVID-19 vaccines. And what we've been hearing is that vaccine development has been very short as compared to HIV vaccines development, and why? That is the question that Dr. Katherine Gill has spoken about. And another question was, COVID-19 has been developed by whites to kill people. That has been said a lot since we started enrolling. That they said why? Because whites are dying more, and now they also want black people to die more. And that has been a big question within the communities. And scientists are used by the politicians to kill people. That is another more statement that is out there. Government never commented on Judge Mogoeng's prayer because they know he is right. That is coming more from the people who are calling themselves prophets, who are associating vaccines and COVID with satanists and demon and all those things. And they said the fact that Judge Mogoeng has said it is because he knows what he knows and he comes from wherever he comes from. Members of parliament must get the vaccine first so that they die first. That is said every day. You hear people talking about COVID vaccine saying that mabafe qala, they must die first. Vaccine is connecting communities with a microchip that is 666. It's an interesting one. I wish I can know how 666 looks like, but people are saying that a lot. And what you also find out is also some staff members within the DTHC are also part of the community with fears and questions. So because we noticed that that is happening, we started with possible solutions. So we had, we would like to have face-to-face workshops with stakeholders, as that would alleviate all what is happening. We also want to have ongoing talks like this involving community leaders. And we need to educate, that came out a lot on Saturday, that we need to educate healthcare workers as they are as confused as general public. We all have, some of us have listened to the healthcare workers unions, they say they do not want vaccines. And now all of a sudden, some say yes, some say no. And mostly that we connect with in our communities, they still say they do not trust the vaccines. And those are the people that we're supposed to trust our lives with as we go to the clinics when we are sick. And then we also need to engage monthly online meetings to clear confusions with the communities. And civil society needs to play part, assisting scientists in clearing out the myths. So that is highly, highly recommended, because civil society can do it. We have done it with treatment and antiretrovirals, and we also need to act now and do something about it. So what happened is last week, MSRS leader had sessions with staff answering all their questions and fears about COVID-19, because also staff had we're feeling I won't take vaccine. Of course, I'm here because I'm working, but it doesn't mean that I do trust what this vaccine is about. And the first training with the committee started last Saturday. That is, it was facilitated by AEDC, MCSJ, and Umoja. Those are some of the stakeholders we are working with in Gugulethu. And then it was very interesting. It came up with, we started with immunology, so that the people understand the immune response and people understand how vaccines are developed, and so that people understand how to engage with bigger communities. So the audience of that session, because it was online for 2 hours on Saturday, starting from 3 to 5, that was facilitated by Dr. Vuiseka Tukula Majola, and Sister Nompumelelo Mantangane was a trainer. She was facilitating the session as well. So it worked very well because the activists as well had fears, they had questions, they are reluctant to taking the vaccines. And after that, all the people that were in that meeting, in that training, registered in that site for taking the vaccines and are willing to communicate with their families. And then that was followed up by the stakeholder briefings that helped to alleviate fears in the community. That happened with 2 churches. Yesterday, Sister Nompumelelo Mandangan and myself, we had 3 sessions with Girls One leadership, and then in the morning, and then in the afternoon, we also had a session with Girls One, like general other leaders for the zones. And it really helped, as much as we picked up that online meetings do not work with stakeholders when you need to give this kind of information. So it means that we need to give a lot of our time as community, like like people working in the science and engaging with community. We need to give most of our time over when people are available, so that we make sure that lockdown regulations are adhered to as we give them the information. And then stakeholder briefings are needed as much as in yesterday. We also noticed that face-to-face are better than online communication. And we are pleading with other like areas as well, because we need more people to conduct these sessions wherever they are. And AEDC Mtonjeni is willing to assist, and DTHC, as they are part of our stakeholders, we are available. The committee team of Emma has been briefed by Dr. Lulu, and now they are clear about the fears that they have, so they're also willing to give the information out to the people. And I am happy today because I said I've got lots of parents and I've got like the participants, and I can't show their faces because of participant respect and confidentiality, but they are happy to be part of the session. And they were very happy when they saw that vaccines come a long way, including what they came here for, the BCG session. Thank you so much. Over to you, Diba.
Thank you, Nombeko. I think with a few minutes remaining, so that we leave more time to discussions, really. I think one of the things, a lot of things that are coming out of Nombeko's presentation is really the fact that we find ourselves in a public health situation where that has everybody's interest in the country, and I guess in the world as well. And I don't know if, I think we know that it is because it is now, this one is not population specific or targeting a specific social class. Everyone is affected by the COVID situation. I think that then is the reason why we have a lot of mistrust and a lot of issues that relate to government and how government is handling the situation have come to the fore. And I think one of the things that we are facing is where the public really associates closely government with research institutions as well as the researchers. And I think the situation has also been made a lot more critical by social media, which presents a lot of great opportunities, but I think in the process, everybody becomes an expert in matters that relate to research and all of this. So what are some of the things that we really feel are important as a result of the situation? Is that as the field that has been doing a lot of HIV research, that has been engaging with communities, as well as engaging with a number of other partners, civil society groups, in really breaking down, like our doctors have done, breaking down the science of health research, and particularly vaccine research. And this is the information that the public is looking for, and the public is looking immediately. We saw that the public looked up to us as the field of HIV and TB that has been doing all of this research. Whilst mobilization and all of the public information efforts have really been happening at levels where a lot of research, where research has been happening in communities, it is clear today that we have to really expand how the information that we are giving out to the communities, so that, because a lot more people are hungry to know facts in the midst of all the misinformation and that is happening out there. I think, in fact, some of the people who question this, they really are desperate to sift between the disinformation that is out there, so that they can understand and really accept vaccine research. And I think this is the work that is in front of us as people who are working with communities and the field of research. So we therefore are required to have a very artistic approaches and techniques, so that we can inform and influence as well as motivate the public and add on to the work that we have been doing. Because at the level of the research that we have been doing for years, I think as Linda-gail has mentioned, we really have achieved a lot in really bringing communities along with us so that they are part of all of this work. Therefore, now we need the requirement at this point is that this public health communication, which has got to be very strategic, and the information has got to be disseminated in a very strategic way. We have got to have mechanisms to evaluate the information that is given out there, if it is accurate, but most importantly, we have got to make information very accessible. And all of this communication has got to be two-way, so that we ensure that we get to a level where society becomes a collective that assures the conditions or where people find themselves in, like we find ourselves in today, that we are able to address them. So I think some of the questions that really come up relate to the processes of conducting research. And I think our two doctors have explained those, that the aspects of ethics and the aspects of human rights protection in the processes of research are very much ensured. And this is happening at a global level, at a national level, all the way to community levels. And those are some of the strong messages that we want to give out to communities. So lastly, what does it take for us to be able to do this? We find ourselves in this situation. It therefore requires a collaboration between government, the research community, as well as community-based groups that are there, that are trusted by communities to be able to give all of the information. And I think, as Nombeko has shared with us, that this is the work that we are doing, but this work has got to be expanded. We've got to recognize the role and limitations of digital communications in the work that we are doing. Because if we have 20 million people who have smartphones, how many are able to access data? So there's a lot of work that we really need to do, but it requires all communities and all society to work collaboratively in order for us to be able to address these the issues that we are facing with regards to to vaccine research as well as vaccine rollout in the public. I think I will end there at this point.
Great, thank you, Ntando. And I think wise words and really helpful to the discourse. There have been a few questions posed, and one that links very nicely to what you've just said is, yes, but why so fast? And why have we, moved at such a pace? And can we be sure that we can rely on the science, given that things have moved so fast? Now, the best answer I've heard to this, and I think each of the two doctors who presented this morning, and both of them were PIs on the ensemble study, the J&J vaccine study, I think can also attest to that none of the actual clinical steps have been shortened. We've done exactly the same that we would have done for a phase 3 anyway. What has been shortened is the time of discovery to making many, many vials of the vaccine for the clinical trial space, then the time to get the protocols written and to get those into the phase 3. All of that has been concertinaed. So the downtime between each of those steps have been eliminated or reduced markedly in order to make sure that we don't subtract time from the actual clinical trial experience. Now, that is very important because we do want to know that the vaccine worked within 14 days, 28 days, and beyond. We now have data on vaccines out to 3 months, and we'll be accruing that data. The ensemble study will run for 2 years. So we will be getting that information over time. So it's important to say to people what we have removed in the timing has been the dead space, the sitting around, taking 2 weeks to have a phone call. We wrote the rollout plan for the 3B of Ensemble literally over the weekend. Now, I promise you that would normally have taken, 6 weeks. We cut it down to a weekend because we feel the urgency. We may have another wave coming to South Africa. We want to get the vaccines out as soon as possible. And so that's our reason for hurrying. I'm going to ask either Katherine or Sheetal to comment if AstraZeneca and J&J have a similar mechanism. Why can we not presume that they would have the same efficacy? It's the $1 million question at the moment, but I want to know, Katherine or Sheetal, if you've got an answer to that. Otherwise, I'll add a few comments as well.
Yeah, so I'm going to just quickly comment on the speed at which these vaccines were developed. I think one of the things that we've actually learned is that we can go much faster than what we did in the past. And I think a lot of our conversations when we look at our HIV work is, sorry, let me just start my video. When we're looking at our HIV work, we're now saying, why did it take us 6 months to do this when we actually can see that we can do it in 6 weeks? Or why can't we move faster? So I think we had all gotten used to working much slower, and the sort of unprecedented need has made us all work a lot quicker. So I think it isn't looking at what we've done, it's looking at why we used to do things so slowly. And yes, I totally agree. I think we didn't jump steps. It was exactly the same. It was the same process, but just we eliminated a lot of the downtime.
Yeah.
And then just quickly to answer, look, I'm not sure why it didn't work, but my understanding is that we don't have enough numbers on the AstraZeneca study to draw these conclusions. And when you're vaccinating a group like health workers that are very, at the front line, you want to plan with a vaccine where defined outcomes, and you've got them in very large numbers. And I think that's what the J&J vaccine gives us. It was conducted when the variant was around, and there were big numbers. And I think we know exactly how it works. So as a health worker myself, I would be quite happy to have the J&J vaccine because it's been trialed during the variant and it had big numbers.
Sheetal, do you want to add anything to that? I think it's an important question.
Yeah, I agree with Katherine. I think it's just a question about having enough data, and I think J&J vaccine has the data to support that it would be better against our variant, and AstraZeneca just lacked that data.
Yeah, so maybe just to step back for folk, it's important when trials are powered, in other words, when we decide on the sample size, we need to know what's the end point, what's the thing we're hoping to see the effect on. Now, for J&J, the company, together with the Operation Warp Speed people, decided, the COVPN network decided that they would power it for severe disease and death. That is something that happens less often. So luckily, thankfully, so it means you've got to really have big numbers, because in order to see something that happens less frequently, you need big numbers to show that. So that is what we get from a 43,000 person study, is we get that critical endpoint of death and severe disease. Now, the study that Shabir Mahdi did on AstraZeneca was done in South Africa, was done on the variant, but it wasn't powered to look at severe disease and death. Now, we know with J&J, we did have a hit on the overall efficacy. The way Shabir showed it with AstraZeneca, the variant is dinging our vaccines as they are today in terms of overall efficacy. But when it comes to severe disease and death, J&J looked the same all over the world, regardless of the variant. Brazil, America, here in South Africa, it didn't matter. We had the same in the mid-80s kind of protection against severe disease and death. So we've got that information in our pocket. We think that may well be something to do with T-cell immunity. Katherine told you about the importance of T-cells. The T-cell part of our immune system may be upholding this aspect of protecting people, reducing viral load of the SARS-CoV-2, and therefore preventing very severe disease that takes you to hospital and needs you to have oxygen. If we can stop that happening, then we won't overfill our hospitals and we won't have all the death we saw in the second wave, which was directly related simply to the fact that our hospitals were too full. So that is the value we would hope we would get out of the J&J. Now, what everyone is saying, AstraZeneca may be able to do that. We just don't have the data. So that is the tricky bit. And I think we're all going to have to grapple with that over the next few weeks, whether we go with our best feelings, our best gut feelings here, or whether we say, no, we have to have the data in hand to know this. And I think that is going to be the trick. That is why I think government has said, let's rather just park it for now. We're having these scientific discussions. We'll come back to the nation about what we're going to do with the AstraZeneca vials. In the meantime, we've got J&J. Let's roll with J&J because we know we've got the data in pockets. I hope that helps. I'm going to ask either Nombeko and Thando. I think this is a good question. Would it be a good idea to get luminaries and politicians and football stars to get vaccinated first in the hope that, not according to your myths, Nombeko, that they would die, but because we want to show faith in vaccines? Do you think that would be a helpful strategy? I'll ask Thando to respond.
Yes, yeah, I see Nombeko is still unmuting. I mean, I think we need everybody. And because we have seen, for instance, politicians in other countries being at least first, and not because, I guess, because they were at risk, but most importantly, to really boost the confidence of people, to really show that, you know, this is something that is safe and something that is actually needed for, you know, for the country. So I think, I mean, that's definitely a good idea. In fact, we do have, you know, other people of influence who are actually saying, no, I am ready. There is a campaign where people are saying, I'm ready and I will take your vaccine. And I mean, if we, for instance, we've heard from Katherine who says she'll be happy to be the first on line to get a jab. So we do need as many people as possible who to really help in sending out a message that this is safe.
Yep.
Definitely. It would be very nice to have people that communities believe and trust more to be vaccinated. And as we say, as activists, that having like our MPs vaccinated first, we are running a risk. And that is what we're communicating to the communities, because should we give it to the MPs, we might as well never get it for the communities again if there's no money to buy more. So rather give it to the communities, let those footballers get the vaccines, let the communities get the vaccines. And we said on Saturday that we are going to fight until we can't fight anymore to make sure that no MP gets vaccinated before communities get the vaccine.
So thanks for that. So Katherine or Sheetal, do you want to talk about why J&J has a single dose and why they went with that versus the 2 shots that is needed for all the other vaccines?
Okay, I'll take that. So always in vaccine studies, there's always earlier research, phase 1 studies, and on the J&J study, they were still trying to decide when we started whether they were going to go with a 1 or 2 dose. But the science came back and said that they were equivalent, and it's much easier to give a 1 dose, much easier to roll that out. And if it gives a similar result, much better to go with the 1 dose. So the safety and the immune response look good after the phase 1 in humans. And so they decided to go with the 1 dose.
Sheetal, do you want to pick up the sort of prime boost concept and then homologous versus heterologous boosting? Because others, and we know that in the UK now, AstraZeneca is being paired with other vaccines.
Yeah, so just simply with the booster vaccine or the second dose of the vaccine, the initial dose would be to prime our immune system against the disease that we're trying to prevent. And then the second dose is usually a booster. So it boosts that initial immune response to create a sort of stronger and longer lasting response to that. To go a bit more deeper would be, I think, too scientific, but that's just simply.
Correct. And now, there's an opportunity to mix and match and to see if, you could prime with an RNA and then boost with one of the viral vectors. So that is work that will quickly unfold now, I think. And we hopefully will get answers along those lines. So again, the immune system is this wonderful, rich array of components, as was discussed this morning. So if you can target different bits of it using different vaccines, you hopefully will get a more robust general immune response down the pike. Now, why we have landed a little bit in the soup with the vaccine so far is that everybody targeted a specific part of the spike protein as the main target for the vaccine's activity. And we know that this miserable virus is mutating even as we speak and is finding ways to get around that target. So it is no longer sensitive to the vaccines that have been discovered. So what is happening now is many of these manufacturers are quickly going back to the drawing board to modify that special target place to say, let's broaden the target so that even if the virus changes itself a little bit, it would still be within the ambit of the vaccines that we'll be offering down the pike. So there is likely to be a lot more movement in this field. We'll be seeing different vaccines coming along, and we'll be seeing different combinations of the vaccines, I think, over time. So if you're given the choice, how will you choose? And I'll round up because we're at the end of time to ask Katherine, how will you choose which vaccine you want to go with?
That's easy. The one that's available first.
Correct. And Tanda, what about you?
One shot looks very attractive, and I'll go for one that really has one shot.
Yeah. Sheetal?
Again, the one that's available.
Yeah.
Put that data on.
Great. Cynthia Nombecker.
The first available shot.
Yeah, so I think you've heard from this team. We are very excited about getting our shot. We want as many of you to feel as excited about getting a shot. It really doesn't matter which one it is, as long as it is shown to be efficacious and safe. And so it has to go through rigorous testing, as all of the mainstream vaccines that have been discussed by Sheetal and Katherine today have done. And so we're in good shape. We just have to get more of them into the country, and they will come. And my best advice is whatever you can lay your hands on, go for it as soon as possible so that you can have that additional protection. There were some questions we weren't able to answer, questions about other diseases, comorbidities, pregnancy. There are many more questions. We will happily be back in this forum to answer more of your questions in the next coming weeks. Please do send suggestions of things you would particularly like to hear about. You can contact Iman Latif, iman.latif at Desmond Tutu HIV Research Center, and she will receive your suggestions or pass them on to Nombeko and Tando or any of the staff at the Desmond Tutu Foundation. Thanks for listening, everyone. Thanks for participating. And of course, thank you to our wonderful panelists. Go well, stay safe, and speak soon.
Thank you, everyone.